Now, let us look at some examples in mammalian systems. Okay, mammalian system gets a little roundabout, but let us try to follow the roundabout here. MAPK, you all know MAPK, mitogen activated protein kinase cascades. MAPK kinase cascades, you know, MAP KKK activates MAPKK, that activates MAPK. That is the way it happens, right? You recall all that. Good. So, MAPK kinase cascades, you all have, you may have seen. These play central roles in responses to stresses, growth factors, hormones, cytokines, and so on and so forth. This also you may recall. This consists of the following: ERK, extracellular signal related kinases, C-Jun terminal kinases JNK, and P38 kinase. These are the three that it consists of. And the principle is something like that: phosphorylation activates relevant proteins, followed by dephosphorylation, which gives you back the original protein. So, that way a cycle gets set up, and then so a process can happen in a cyclic fashion, which is what makes a continuous processing possible in the cell. Right. As mentioned, MAPKK activate MAPKK, MAPKK activate MAPK, and so on and so forth. And ASK, which stands for apoptosis signal regulated kinase 1, ASK1, is a MAP KKK that regulates JNK and P38. This ASK1 can regulate JNK and P38 here. And ASK1 is activated by stresses. So, stresses cause reactive species formation activate ASK, and this process can happen. We will look at some details. Might want to write this down. Gives me time for some attendance. Today it's a lot less. Okay, I guess it's getting closer to the 2-month mark. I typically have this for 2 months after the cold. Okay, some details of ASK. ROS directly affects the kinase ASK1, and the process is something like this. You have a dimer which is inactive, which has TRX here, that gets released when the protein gets oxidized, and when the TRX gets released with the disulfide bond formation, the disulfide bond formation is the oxidation, and that releases the TRX, ASK becomes active. So, now it can do whatever we mentioned earlier, activated by stresses in this fashion, and then it goes forward and does its work. Okay. So, ROS inactivates this is another example. Okay, that example just to tell you how it worked and so on. Oxidation released, then it becomes active. This is second example. It inactivates, right? Yeah, dephosphorylation of the as an example PTP, protein tyrosine phosphatase. And what does this do? It does something like this. It's a busy figure. Don't worry too much about it. Let's focus on the signaling protein here, which is inactive. Okay, the signaling protein here gets activated by tyrosine kinases, which come about because of growth factors, cytokines, and hormones, and so on and so forth, and it gets into an active form. So, this is the active form. When the job is done, the PTP converts the active form into an inactive form, right? So far, so good. What happens when ROS plays a role is that it modifies this PTP. It modifies the PTP and converts this form into a sulfenic acid or a sulfinic acid or a sulfonic acid or other forms. Many things can happen. When this happens, this becomes inactive. PTP becomes inactive. When PTP becomes inactive, it cannot convert the active form back to the inactive form. If that is the case, this gets stuck, right? And so, that is how the PTP plays a role in affecting the process.
So transcription cannot happen due to the inactivation of
No, the conversion of this form to this form, the that's the enzyme that converts it. Nothing to do with DNA. Just an enzyme. The enzyme has to be active for the reverse process to happen. It's not an active enzyme, therefore the reverse process does not happen. Get stuck there. Okay, so that is good enough. Busy figure for a different purpose altogether. This is from a different paper. The purpose was a lot more elaborate. So we'll just look at the ones that we want. Okay, so this is another example. Third example is PI3K pathway. Most of you would have heard this PI3K pathway, phosphorinositide 3 kinase pathway. It plays a role in many different important processes in the cell in mediating the effects of growth factors and insulin, for example. Phosphatase and tensin homology phosphatase, phosphase and tensin homology phosphatase, P10 for short, is an important negative regulator of the PI3K pathway. In other words, if P10 is inactive, then PI3K is active. Okay, so that is the molecular biology of things. When oxidation happens, reactive species, for example, H2O2, studies have been made with H2O2. When oxidation by H2O2 happens, this oxidation inactivates P10. We already know how it can do that. It inactivates P10, and therefore that activates the PI3K pathway. And the reverse is true. The reduction of P10 inactivates the PI3K pathway. So you have both happening, and therefore a series of processes happening, and therefore signaling happening. Signaling being caused by something.
So usually when we study pathways, it's always the enzymes that we remember and keep in mind. So all these diagrams that we see, they don't have much enzymes, only we see the.
This phosphatase is an enzyme.
I mean, does it mean that the ROS actually acts on the enzymes and in turn
We have looked at examples where it acts on the proteins. Protein could be an enzyme. Protein could be an enzyme, protein could be a transcription factor, and so on. So wherever it can, it can oxidize many different things, but at the signaling, it has to operate in a narrow range. That narrow range, these are the things it does, and this is when it is working fine, when the cell is working fine. Go a little beyond, then it can oxidize many different things, and the whole thing falls apart. So this is the homeostatic working regime of reactive species, and finely controlled. And although it is highly reactive, it can play a significant role in cell signaling, which is a coordinated process. Any other questions, comments? Okay, one more example, another popular one, NRF2. And in this case, we are going to look at NRF2 ARE. NRF2 stands for nuclear erythroid 2 related factor. ARE stands for antioxidant response element pathway. Okay. So they go together, NRF2 ARE. That's an important pathway in the defense mechanism against oxidative stress. So if oxidative stress causes damage, this pathway is one of the things that helps in recovery. It controls the expression of genes that produce proteins which act against oxidative toxic substances produced by ROS interactions and affect detoxification. That's the way they work. That's the way this pathway works. ROS oxidation of a complex complex of proteins KEAP, CUL3, NRF2. That releases NRF2 from it. This is the way it happens. It initially, the NRF2 is initially a part of this complex KEAP, CUL3, NRF2, and the oxidation releases NRF2. Okay, that's where it starts going. The complex is originally in the cytoplasm. The NRF2 gets into the nucleus, binds with ARE, which is in the nucleus. ARE is the antioxidant response element protein in the nucleus, and that gets further activated by other oxidized proteins. Lot of things happen after that. ARE is a transcriptional factor for many genes with antioxidant functions. Okay, so you have this complex, oxidation by ROS releases NRF2. NRF2 goes into the nucleus, binds with ARE, and then starts off the transcription of various genes that produce proteins with antioxidant function. That's the way this works. Okay, simplistically speaking, there are a lot of details. It is not easy for me to read through this to extract this out, because the biology books will give you all the details. The big picture you will have to, big picture for our own purposes, of course. Purposes are different. Okay. Any questions or comments? So, we saw some examples with bacterial systems, we saw some examples with mammalian systems of the ways by which the reactive species can affect cell signaling. Right? They modify proteins effectively. Different kinds of proteins have different functions, and that's what happens, and it can happen in a coordinated fashion. This is used to control various things. And by the way, you'll soon realize that oxidative reactive oxygen species are the ones that mediate many different stresses. It could be exposure to a chemical that we have seen so far, HCl, H2O2, and so on and so forth, so far in the course. It could be heat, it could be light, it could be pressure, it could be temperature. All these stress effects, it could be shear, physical shear stress. All these shear effects get mediated through reactive species. I can make this statement today with a lot of confidence. Earlier, we were saying we believe this happens for the past 30 years or so. We've been rather, let's say, for the about from the late 90s. That's when I got into this thinking. Late 90s on to even late 2020s, 2010s, and 2018, and so on and so forth. I had to say we believe that this happens, and now people accept it. And we contributed to that acceptance. Right? We have done studies over with various different stressors, and we have looked at the quantitative reactive species extents, and that's why we say the reactive species is the best indicator of the stress levels in cells. Not easy to measure. I mean, as of now, the dyes are expensive and so on and so forth, but these pseudo steady state levels of reactive species are the best indicators of the stress levels in cells. That was, I mean, we had contributed to that thinking. So, we saw effects at pathway levels and so on and so forth. It can also have effects at systemic levels. I'll give you 3 examples. I'll dwell on one, and or I'll give you some details on one. The other 2 you can look it up. I'm going to talk a little bit about this paper by Finkel in 2011. This was in Journal of Cell Biology. We're going to talk about the initial Warburg effect, and Jayashree can relate to that. Chen et al. 2018, British Journal of Pharmacology. This shows the effect of reactive species on vascular diseases, a systemic effect. And Yang and Lian in 2020, Molecular and Cell Biology. Take a look at table 2, which lists a lot of these effects, systemic effects. Okay, so just write these down. The first one I'm going to talk about in this class itself, and then the other 2 you can look it up. Okay, let's look at some parts of the first paper. That is a nice story there. Okay, this is signal transduction Bioreactive oxygen species by Torrent Finkel in JCP Review 2011. One person is from Center for Molecular Medicine, National Heart, Lung and Blood Institute, NIH, USA. I am looking at the introduction part, just the initial introduction that will tell you. Let me read it out to you. Some more. Yep. Are you able to read? A little bit more. Little bit more. Lot more. Okay, I'll read it out to you. No, I can go. That's not a big deal. Is it somewhat better?
Yeah.
No, it's better. Okay. Let's start with the introduction here. Don't worry. The abstract is about the entire paper. We are not interested in that right now. Very early in his career, while working in a remote marine biological lab in Italy, the young Otto Warburg observed that fertilization of sea urchin eggs resulted in a rapid and early six-fold release in oxygen consumption, published in 1908. The notion that oxygen consumption was dynamic and seemingly tied to cellular proliferation would have a profound effect on the young scientist. Later, these ideas would be refined and focused not on normal fertilization, but rather on the metabolic abnormalities of cancer cells. Some 23 years after his observations in Italy, Warburg was awarded the Nobel Prize for his discovery of the nature and mode of action of the respiratory enzyme. Interestingly, although Warburg's oversight observations have been confirmed by many others, his hypothesis that this represented a burst of mitochondrial oxidative phosphorylation is undoubtedly incorrect. Okay, this is a Nobel laureate. Okay, that's perfectly fine. That's science. Indeed, nearly 100 years after his initial observation, it was established that a surge of oxygen consumption after fertilization is not as originally envisioned, some primitive metabolic wake-up call by the young zygote. Rather, it would seem oxygen is instead used by specific NADPH oxidase on the egg's surface for purposeful production of nanomolar concentrations of hydrogen peroxide. This was shown in 2004. Surprisingly, this burst of hydrogen peroxide production does not damage the nascent organism, but instead is used as a part of an enzymatic reaction that ultimately results in the development of a protective shell around the young fertilized egg. These observations are in concert with a growing body of evidence that suggests that ROS growing body of evidence in 2011. Now it is reasonably established. That suggests that ROS can be purposefully made and harnessed to regulate a diverse array of physiological processes. In turn, accumulating evidence also suggests that dysregulation of oxidant signaling may cause or accelerate a host of pathological conditions, including the rate we age. Thus, seemingly from life's inception to its end, redox signaling acts as an important regulator of physiological and pathophysiological outcomes. Herein, I will try to selectively review some of the highlights and emerging trends in oxidative signaling. If you are interested, you can go and read the paper, you have all the details. Okay. So this shows a systemic effect, developmental effect, directly role played by reactive oxygen species, production of hydrogen peroxide and so on. And peroxide is a very important molecule. This does a lot of things. Not too reactive, so that way it has the time to do a lot of things and so on. So this is where we finished this chapter. We have finished three big chapters, four chapters. The fourth one is not too big. 4 chapters in the course. I think we are in good speed, not bad at all. So I'd remind you to start looking at your RS leverage exercise a little more carefully, get your thoughts done. Okay. People who have done CFA know that what a creative process is. It will turn you inside out, but you know, it's a horror when you go through it, but so elating when the final thing comes out and the learning is so much greater. Okay, for any creative process, right from the painter to whoever does it, this is what it is. Talk to any artist, they'll tell you this. Okay, so this is nothing new. And in this case, it is being used to learn things in the best possible way at the deepest level possible. Okay, that's one. And then start looking at your papers. It's a more standard exercise, but it will help you focus your thoughts on one aspect of things that are already available in the literature in a critical point of view. Okay, those two are going to contribute.
Deadline is at the first week of May.
I'll tell you the deadline for submission of the leverage exercise. It will be a week before the last day of classes, so that it will give me time. It takes me a lot of time to grade them and give grades on time. So I asked for a week before the last day of classes. You will present your critical literature review in the last 5 classes of this course. Okay. I will tell you the exact dates and who's going to present when. I'll do that in class also. I'll draw lots essentially. And so if you have time, you can do one. No, one is a little too little. So 2 per class. 2 per class. So there are 11. So we need 6 classes. Leave alone the last class. I usually have 1 buffer class at least. Leave alone the last class. We'll do the exact numbers later. And then the 6 classes before that will be presentation classes. And sometime during that, you'll have to submit your leverage exercise also. So you start working on it and so on. So that, and from now onwards, there will be a change in the way we do things. There will be papers that are available to you on your Moodle page. Some of them are required reading, and some of them are optional reading. Okay, if you're interested in that particular area, you'll be interested enough to read the optional ones also. When you come back to the next class, I'll give you time to read, by the way. When you come back for the next class, there'll be a incentive quiz, okay, for lack of a better word. I don't like the word quiz, but incentive quiz, where it's open notes, open book and all that. As long as you know, you've read things, you'll know where things are, you can easily do well. On that, just to make sure that you've read through the required papers. I went and just opened up the first set of papers for you. On Moodle, it's oxygen supply and reactive species. There are two papers to read only. To start out, we'll start out simple. I mean, it will take you time to read, of course. And there are 4 optional papers, 1, 2, 3, 4, and all of them are R papers. Okay, you can get the. I think I've given you the PDFs here, so you don't have to worry. If you want, you can get the details from my website and directly go to the journal also. But since the PDFs are available here, that may not be necessary. So you read these papers. I'll give you time. So I'm assigning it today. We will not have the next class. So we'll not have a class tomorrow. Okay, there's no class tomorrow. We will have the how should we do it? Come prepared on Monday. Depending on how things go, we may have it on Monday or we may have it on Thursday. Okay, I'd rather continue a little bit at least on the story the day I give the day you're fully prepared, the day you remember everything and so on and so forth. Let me say how it goes. We will have a class on Monday. There's some holiday coming up, right? When is it? Friday. Okay, the next Friday. That's 6th of March, isn't it? 8th of March. Okay, then fine. Then don't worry about it. So we may have the incentive on Monday or on Thursday. Come prepared for both these 2 papers. You can go and take a look at your Moodle page. The papers are there. So that's the way it's going to be for the next few things. You're going to read those papers, so you'll have some sort of a background. And then I will give you some presentation. It's going to be more of a discussion. You will raise questions and I'll answer. It's entirely on our work. The course shifts to our previous work over the past 3 decades. Almost, yeah, 3 decades. In 93 years. 93, I started my independent research career. Manjula joined in 96 Jan. So we had gone in this direction in late 94, early 95 itself. So around that, about 3 decades of work in as interesting a fashion as possible. It will be in the form of stories so that you can discuss. But read the paper so that you'll have all the details and raise questions with them when you come to class. That way, it will be a good discussion. Okay. Any questions? Not. We have a minute left today. Good. I'll meet you on Monday, noon time, and then we'll take